So our next speaker is Sandeep Kumar. So Sandeep, well, hardly needs an introduction, I think, because he is one of the, let's say, like one of the most prolific developers of antibody computational tools. Like he contributed to many, not only computational tools, but also like actually to creation of this field, and of course, like as it goes to development of several biologics currently. He used to be at Boehringer Ingelheim, but currently he is continuing his efforts at Moderna. Yeah. So today we're going to hear about Sandeep talking about biopharmaceutical informatics. And Sandeep, the floor is yours. Thank you very much for coming.
Thank you so much, Conrad. Can you hear me all right?
Yes, I can hear you, and I can actually see your slides.
Okay, thank you. So thanks, everyone, and thanks, Antibody Society, for asking me to present about biopharmaceutical informatics and how we can actually use computation and experimentations in a syncretic way to facilitate discovery and development of biotherapeutics. I should say really at the very beginning that what is really our goal as pharmaceutical scientists? Our goal is very simple. We want to bring medicines to patients that are effective, that are affordable, and we want to do it fast. I think biopharmaceutical informatics, I came up with this vision mainly keeping these three goals in mind. Therefore, I actually call for really using computational tools, experimental data, molecular simulations, and machine learning together in order to kind of understand and find solutions for the current challenges in discovery and developments of biologics, but also inform as the projects progress along the discovery to early discovery, early development to product development to clinical stages, and ultimately commercial, to inform, to enable a data-informed decision-making at each and every step of the drug discovery. And I kind of feel that biopharmaceutical informatics can be used in both ways. One could be the incremental innovation, which is basically transforming and digitizing and digitalizing as much as possible the pharmaceutical industry data that we have and taking advantage of being able to develop newer and newer machine learning and AI tools. And the second is, of course, the disruptive innovation where we can say that can we actually discover antibodies and bring the antibodies to market with minimal experimental efforts. And really then if we really think in those terms, the question is that what is the excitement? What is the currently available digital tools can help us with? And I think the first important part over there is that can we expand that druggable antigen space? Can we build antibody libraries? Can we build repertoires against novel targets that are hard to develop antibodies against in conventional ways? And I think that this is where a lot of therapeutic values for the current excitement lies in my mind. Then second is, can we actually improve or can we make our products, work products or the projects go faster and in a better way? And can we actually start using the technologies in such a way that folks in the lab, that people who work on the bench, they can actually start bringing the technology and their effort is reduced in the sense that they don't have to do experiments by trial and error. They can focus more and they can actually, in a way, within the same time period with the same resources, achieve better. Second, of course, is that can we improve the clinical success rates of which, of course, almost everybody knows that for biologics, it's probably 1 in 5 these days. For small molecules, it's more like 1 in 10. But that is for the antibodies that reach the phase 1. If you actually looked into antibodies or formation of novel therapeutic concepts, then the rates are much lower. So what's the opportunity more in terms of in silico antibody development? And I would say that if you look on the left-hand side is the conventional broad overview of the discovery processes for biologics. We start with antibody generation in animals in via immunizations, hybridomas, display technologies. Then we look for functional antibodies from there, the antibodies that bind to the antigen of our interest. And then we actually ask the question, are they developable? Are they actually something that we can make a drug product out of it, something that I can commercialize? And of course, everything takes time and money and costs associated with that. But if we think about discovering antibodies in silico, we could think about generating antibodies using AI, ML techniques, antibodies that could be basically creating or getting inspirations from antibody repertoires, which could be naive or antigen specific. So in a way, we could generate libraries that potentially could be even bigger than the libraries or the repertoires that are seen in the humans or animals, both in antigen agnostic and antigen specific ways. And then what we could do is that if we have our good antibody profile based methods done, then we could actually pay for developability upfront. And we will still be left with enough number of antibodies space or enough amount of antibody space that we will be able to still select antibodies, and typically that bind a given antigen. And I think that's where the field still has to evolve a little bit. And here we can ask the question, can we actually virtually screen antibodies against a given antigen, find the potential binders, dock them, and then use that docked definitions of antigen antibody complexes to affinity mature them. So in some ways, can we capture the whole process of discovery in silico, but also improve on it by paying for developability forward? And I think we are, in some ways, the matter is that in isolated ways of this whole concept or roadmap for an in silico antibody discovery, we are already there. And it's a question of being able to synthesize them and make the protocols and workflows much better. So for example, with the help of our postdocs, Nandini, and also Udit and others, we were able to generate using GAN methods, the antibodies completely in silico. And on your left, the antibody model that I show you is basically is not a natural antibody. It is an antibody made by AI. And because we had pre-selected them for their developability and for their medicine likeness, we found that in experiments that they did possess good developability profiles. And now comes the question about virtual screening. There are several methods for virtual screening, and a lot of these methods can also be inspired from small molecule world. Pietro showed an example of fragment-based screening and design of CDRH3. We took the pharmacophore-based approach, and we actually were able to create pharmacophore out of epitope of a given antigen, and were able to find the antibodies that would putatively bind it. And then, of course, this is a structure or complex that can be further used to improve the affinity of the antibodies using computational design. And this actually was also shown in a third part where in one case of a project, we started with an antigen antibody complex, and we were able to design the libraries just based on mutating the paratope residues for different affinities. And we were able to put them on a phage display, and we were able to get fine antibody binders with varying affinity, as well as in this particular case, we didn't really have a cross-reactive antibody. We were able to actually start with the human antibody and make it more cross-reactive to cyno. So in some ways, what I have shown over here to you is really that discovery of antibodies in silico, as I call it as DABI, is actually, we have realized this vision. And of course, others in the field have also the examples where they have realized this in parts. The question is, can we really make it unified, and can we actually make it more algorithmic so that this process then can be more and more automated? And we reach to the stage where experiments are done not for trial and error, but rather to confirm that we are on the right track. And I think now I'll switch gear more towards the digital transformation. This is a big issue in pharmaceutical industry where we have lots of data, but that data resides in people's presentations, Excel sheets, lab notebooks, but really not into the databases, but that are fair and findable and usable by data scientists, along with, of course, the experimentalists. So I think there are huge opportunities for us to use technology. In fact, I often say that we use technology much more in our homes than we do it in our labs. But what is really the strategic imperative for digital transformation? Why should we do it? And I think that I would argue for three major reasons we should be doing it. We should gain process efficiencies as much as possible, reduce the time and effort, and accelerate the discovery and development process. We should be able to learn from the data that we have, if it is available in self-consistent manner, in a fair manner, to enable technologies based on machine learning so that we can create the digital twin for each process. And therefore, we could essentially predict reliably before we actually do the experiments. And then we can choose from those predictions where or which candidates to test in the lab. And I think that major part of all of these things is that this transformation is patient-centric in the sense that it enables us as a pharmaceutical scientist to bring better medicines to patients faster and in a more affordable way. And I think that's the major, how should I say, boon for digital transformation. And to really kind of overemphasize the point in a way, is that digital transformation essentially works or has basically three stages. One is the digitization of the data being in the databases. Second is the digitalization, where we are creating digital twins. We are creating models, both AI models, molecular simulations, to connect the molecular properties of our drug candidates with the experimentally observed properties. For example, biophysical experiments. How can we connect the microscopics to macroscopics? And the third is, of course, really transform the whole process. And I think the benefit for digital transformation increase as we go along the chain, but the cost is actually more right at the digitization stage and the digitalization stages. So it's a balance between cost and versus benefits and efforts that are required for this. There are, of course, when we think about it, how do we do it? This is what I gave you was a very broad, high-level overview. So how do we do it in reality? And I think for that, we have to really consider different stages of drug discovery process. Essentially, first is the reagent or the antigen expression purification, and then generating antibodies against those antigens, selecting antibodies and finding a lead candidates, and then eventually optimizing the lead candidates for developability. And there are several considerations at all of these stages. And I think there are also several computational opportunities in these stages. So for example, we can easily now predict antigen structures. Most of the time we do have structures available in the databases, or we have AlphaFold models. We could engineer the antigens that are poorly behaved for better expression and purification in the lab in such a way that it does not really affect their immunogenicity. We can construct in silico antibody libraries, something that I spoke about DABI in the beginning, and or we could actually redesign the CDRs of a highly developable antibody to bind the target of our interest. We could profile and pre-select antibodies with good developability, and then therefore reduce the number of experiments required to identify a lead molecule. We could also map the epitopes and paratopes in a better way. And many times, if we can't find a lead molecule within a given germline pair that gives us all the properties that we need for a drug project, we can actually use consensus protein design to go forward with it. So there are several opportunities over here, and I would not belabor this point further, but I'll go into some of the work or credit. The reason we need to do this is that we need to talk between discovery and development data in a cyclical manner, and we should learn both from discovery and development. And eventually, so to show that some of the things that we have come up is really the medicine likeness profile. I think Victor mentioned about the 5 parameters that we were able to identify, and for the intrinsic physicochemical profiles by analyzing the data on antibodies that are currently medicines. They are not in clinical trials, they are not clinical stages, they are medicines that they are available right now to the patient, so they have been all approved. And then we found that these profiles can become even much better in being able to discriminate for developability, which in my mind is of course not just manufacturing, but it's also safety, efficacy, pharmacology, all combined together. So we were able to put the dynamics into it, and we found that with the dynamical effects, it's actually performs much better. We had also worked on, and I'm here giving you a few examples from our work. We have been also able to essentially analyze antigen antibody complex crystal structures and map the paratope residues and onto multiple sequence alignments of the antibodies. And we could show that these paratope residues fall at certain positions, and even the hotspot falls on certain common positions. And this effort allows us to then predict the antibody paratopes right off the bat as the sequences come out from antibody generation campaigns. And this is some of the examples where we compared our predictions with the crystal structures, where in the orange are the paratopes predicted, identified from crystal structures, and the green are the predicted paratopes. And I think we are able to recapitulate the crystal structure paratope predictions based on the sequence and the structural modeling of antibodies candidates. We also used, in collaborations with Pete Tessier and others, we are also able to lay our hand on large number of NGS data and use that to really identify physicochemical attributes for reactive antibodies that bind non-specifically. And we found that for the variable regions, region PI3D, and the positive patches in the CDRs are one of the major discriminatory factors for polyreactivity. We want the PI to be high for the case of to develop highly soluble antibodies, but we don't want it to be too high that antibodies start binding the receptors in a very non-specific way. So like Pietro had said earlier, this is a trade-off. And then the idea is, can we actually go from monospecific antibodies to multispecific antibody formats? And how do we combine antibodies for different specificities? It is a little bit easier for monospecific antibodies where both the variable regions are really identical or very similar in their 3D shapes and everything and properties, while in the multi-specific formats, they can vary a lot. Again, we're using very simple ideas around percent monomers and interpreting in them in terms of germline pairs and their compatibilities. We were able to make a few digital tools around it. And you know, the opportunities for computations are not limited only to the discovery. In my Pfizer days, I used to be in formulation development and the drug development part of the organization, and where we started actually thinking in terms of how can we apply computation in a place which is extremely experimental, extremely hands-on. And we found that there are, again, a number of opportunities where computation could help us identify and forecast the developability challenges. One major reason is very simple. If you look at any biotherapeutic drug that goes under development, product development stages, it goes through during its manufacturing, storage, shipping, it goes through many stresses. But at the end of the day, the consequences of these stresses, and most common of them, is actually aggregation. And we started this work back in 2009, and we started looking into aggregation-prone regions in marketed biotherapeutics. And we essentially use the same type of algorithms, Tango, Page, that are used to identify amyloidogenic regions in proteins, in intrinsically disordered proteins. And again, we found that these APRs are found at specific positions in the antibodies, and typically those which are in the framework regions are buried and therefore tucked away, and they are not at developability risks. But sometimes the APRs that lie at the CDR and framework interface, or like in the Vernier regions, that could be quite deleterious for developability. And I think I have only a few minutes left, so I'll try to go fast. But I think the one part we actually saw was back then was that we could actually correlate aggregation that we used to observe with immunogenicity of the products. And most of the time people used to say aggregations can be immunogenic, but how is it that? And we found that aggregation-prone regions often tend to be part of T-cell immune epitopes. We were able to see this in case of biotherapeutics, but then we actually later on expanded this to amyloidogenic proteins, and we started analyzing the data on the protein sequences that are involved in neurodegenerative diseases. And we again found that aggregation prone regions in these proteins are also part of the immune epitope. So this is one of the examples where the drug development actually fed into research that's useful for drug discovery. And I think after this, I would like to just talk about one more thing, which is to predict the colloidal stability of antibody solutions. And I think this can again be very easily profiled using the variable regions of proteins. And essentially colloidal, we want largely repulsive solutions of antibodies. And that can happen if your variable regions are positively charged, because most of the constant regions at the typical formulation pHs are positively charged. And so now, depending upon the PI of the variable regions, we could run into highly repulsive solutions versus solutions that could actually be highly attractive and therefore have low colloidal stability. All of this is very theoretical, very conceptual work, but it also was, we practically demonstrated it in terms of an antibody, which was led off the product development because of several developability challenges. And essentially, it took us only 2 amino acid position mutations, one to disrupt an APR, the aggregation prone region, and another to disrupt a highly solvent exposed negative charge patch. And this led to considerable overall improvement in the developability of the antibody. Of course, the project was dead, but we could show that our variant was much better in terms of the biophysical properties. So in my last slide, or last but one slide, I just want to kind of say this as a take-home message that biopharmaceutical informatics is an emerging field that has a lot of promise, and it can make the biotherapeutic drug discovery and development more efficient, faster, and more cost-effective. With this, I would like to thank my postdocs. Nandini contributed a lot towards discovery of antibodies in silico and epitope paratope predictions. Lucky came up with a medicine likeness profile, which of course was further refined by Kyle Martin and Giuseppe. All of these postdocs actually are now full-scale scientists in different companies. And then of course my BI colleagues, Kenny Sang, my supervisor Andy Nixon, my collaborators Mike Marlowe, Yosh Kabar, and Jan Kriegel. And of course, I'm also blessed with collaborating with academic labs, especially Peter Tessier, Victor Batista, Charlotte Dean, and Michael Gromia. Thank you very much for your attention.
Great, Sandeep. Thank you very much for this talk. I think that there were 2 questions asked by the same person, but someone from the audience kindly responded to one of them, so at least one. Could you suggest some tools for affinity maturation?
Yeah, so we essentially used just the residue scan in Mo. So we started with the crystal structure of antigen-antibody complexes, and we started going with the protein design module in Mo, and we first identified what are the most hotspot residues by doing a alanine scanning, and after that we started exploring those positions with different amino acids. And that helped us create a library, because these calculations are very good at single point mutations, but combining mutations to get a combinatorial effect is still a challenge for any of the free energy calculation algorithms. So we actually put them on a phage display library and panned them for higher affinity variants.
Cool. Thank you very much. So maybe one more. In your analyses, did you observe an overwhelming contribution of CDR3, presumably H3, to antigen binding, or do you see other CDRs that have been leveraged for affinity and/or developability?
I think, yes, the common notion in the field is that heavy chain CDR3 contributes the most. But if you really analyze antigen-antibody complexes, you would find that even heavy chain CDR2 and the light chain CDR3 also make significant contributions towards antigen binding. And in fact, heavy chain CDR2 and heavy chain CDR3 make equal contributions in most cases, except that heavy chain CDR2 is really long compared to heavy chain CDR3. So when you look at per residue contribution, heavy chain CDR3 trumps almost every other CDR. But there are cases and there are examples where actually the heavy chain CDR3 is not even involved in the binding, and binding of the antigen is coming from CDR2 or CDR3 in the light chain, or even sometimes CDRL1. So I think the nature is very versatile and very resourceful, I would say. And it's we usually think that heavy chain CDR3 is the holy grail, but I would say that there are examples where it's not the case.
Fair enough. Super, Sandeep, thank you very much again. I think we're going to move on to the last